Conditioned fear is typically studied in humans by pairing a neutral stimulus, such as a tone, with a sudden, loud burst of noise. With repeated pairings, the tone elicits a startle response, which is measured by electrodes placed on contracting muscles over the body. Conditioned emotional responses in this paradigm can also be measured as changes in heart rate, blood pressure, and galvanic skin response. The same responses can be measured in humans. Navigating through a virtual environment containing snakes, spiders, or other aversive stimuli, see Figure 4.5. Figure 4.5, schematic of a virtual reality setup to test conditioned fear in humans. Conditioned taste aversion. Many people feel nauseated when they smell or taste a particular food. This often reflects a conditioned taste aversion, CTA, that developed when consumption of the food was associated with gastric illness, usually vomiting. It does not matter if the food itself caused the sickness. A coincidental food nausea association produces a conditioned taste aversion. For example, an individual who eats a banana when they have a stomach virus may later have an aversive reaction to the smell and taste of this fruit, even if it was not the banana that made them sick. Conditioned taste aversions are easily observed in lab animals. After consuming a flavored substance, animals are made sick by injecting them with a nausea-producing agent, such as lithium chloride, or exposing them to low-level gamma radiation. When they recover from the illness, animals are presented with the flavored food. If animals eat less of this food than another food that was not associated with illness, they have developed a conditioned taste aversion. Conditioned taste aversions can be very powerful. They often develop with a single conditioned stimulus-unconditioned stimulus pairing and are sustained for long periods of time. This should not be surprising, as poison avoidance learning is critical for survival. Animals can become very sick or die if they consume inedible items, so they must learn to avoid poisons and other toxins by associating the smell and taste of the food with illness. This is one reason that it is so difficult to develop effective poisons for rodents. After sampling a very small amount of the novel food, rats and mice feel ill and avoid this food in the future. Note that the measure of classical conditioning, the conditioned response, varies across these three types of paradigms. Given that there are dozens, or more accurately hundreds, of other classical conditioning tests, the number of ways in which scientists can measure this process is almost endless. In addition, for each conditioned response, a researcher may choose to measure how quickly it is acquired, how large the response is once conditioning has occurred, and how long it lasts when the unconditioned stimulus is no longer present. This sometimes makes it tricky to compare the magnitude of conditioning across studies. Researchers, and students reading these studies, must pay close attention to the dependent measure in each study, the Y variable, because discrepancies in research findings can sometimes be explained by differences in how the conditioned response, the Y variable, is assessed. Operant conditioning. As noted previously, contemporary studies of operant conditioning can be traced back to Thorndike's experiments with cats. These animals were required to pull a rope, move a stick, or push a board to escape from a puzzle box. Operant conditioning, or trial and error learning, as Thorndike called it, was evidenced by a decrease in the escape time over trials. Discrete trials. This discrete trial setup, in which subjects have the opportunity to make one correct response for each time they are placed in a testing apparatus, is still common in lab-based research. The water maze, T-maze, and straight alleyway are all discrete trials operant conditioning paradigms, although most of these tests are used to assess other cognitive processes such as spatial learning or decision-making. A simple example of operant conditioning using discrete trials is the conditioned avoidance paradigm. In one version of this test, a rat is shocked for stepping off a platform. Longer latencies to step from the platform on subsequent trials indicate better conditioning. Unlike the conditioned freezing paradigm described above, the presentation of the shock in the conditioned avoidance paradigm depends on the animal's behavior. If it does not step off the platform, it will not receive a shock. In classical conditioning tests such as conditioned fear, the unconditioned stimulus follows the conditioned stimulus regardless of what the animal does. Free operant. When most students and researchers think about operant conditioning, They imagine a rat pressing a lever for food in a small chamber. This setup was designed by Skinner in the 1940s as a means to evaluate the ongoing responses of his subjects, usually pigeons or rats. Animals are placed in a chamber containing some manipulandum, usually a lever, which can deliver a food or sugar pellet. In contrast to the discrete trials paradigms, free operant methods allow animals to respond repeatedly, freely, once they are placed in the experimental chamber. One advantage of this method is that the experimenter can observe variations in responding across time. This information is represented on a cumulative response record, such as those shown in Figure 4.6. The y-axis of the graph represents the total number of responses in the session, and the x-axis indicates time. Thus, the cumulative record provides a visual representation of when and how frequently the animal responds during a session. Figure 4.6. Cumulative response records for rats responding for methamphetamine, A and B, or food, C and D. Food reinforcement produced a steady rate of responding over time, whereas methamphetamine produced a step-like pattern in which a drug infusion, i.e. presentation of the reinforcer, was followed by a pause in responding, A. When animals are responding for drug, they load up at the beginning of the session, presumably to attain an optimal level of drug in their system, A. This effect is not observed when animals are responding for food, C. Administration of the glutamate antagonist, MTEP, B and D, led to a near-complete cessation of methamphetamine reinforced responding, B, but had no effect on responding for food, D. The rapid lever presses that occur at the beginning of the methamphetamine session, B, indicate that animals are expecting a drug infusion and may be frustrated by the lack of drug effect. Animals stopped responding for over an hour, tried the lever a few times, and stopped responding for another 40 minutes. This pattern is consistent with the idea that MTEP is blocking the reinforcing effect of methamphetamine. Okay, so we have 4 graphs. The top 2 are about methamphetamine. So we have time from 0 to 120 minutes on the x-axis and number of responses from 0 to 40 on the y-axis. Okay, so in A, the rats are trying to load up on methamphetamine the most at the beginning. You get a step-like pattern because they get the drug infusion, then they pause responding, which doesn't happen with food, which generally increases. You can see that in C, it's almost linear. Okay, so in B and C, glutamate antagonist MTEP is administered, which makes the rat stop pressing the lever for more meth. But as you can see in D, the rats, even though they have the glutamate antagonist MTEP in their system now, they will still press the lever for food in an almost linear fashion. Okay, and you can see in B, because they're not getting methamphetamine at the beginning, they press the lever a bunch of times because they're waiting for the methamphetamine infusion, and they're frustrated because they don't get it. And they don't keep pressing the lever because they're injected with MTEP, and that is blocking the reinforcing effect of the meth. In free operant paradigms, the relationship between responding and reinforcement is described by the reinforcement schedule. This is a rule, set by the experimenter, that determines how and when a response will be followed by a reinforcer. When every response produces a reinforcer, the schedule is called continuous reinforcement, or CRF. More commonly, responding is reinforced on a partial or intermittent schedule. One of the simplest ways to produce partial reinforcement is to require animals to make a certain number of responses for each reinforcer. If the number is set, the schedule is called fixed ratio, FR. If the number of required responses varies about a mean value, the schedule is called variable ratio, VR. Thus, fixed ratio 5, fixed ratio 10, and fixed ratio 50 schedules require animals to make exactly 5, 10, and 50 responses before the reinforcer is presented, whereas variable ratio 5, variable ratio 10, and variable ratio 50 schedules require animals to make an average of 5, 10, and 50 responses. A progressive ratio, PR. Schedule is a variation of a fixed ratio schedule in which animals must make an increasing number of responses for successive presentations of the reinforcer. Typically, the schedule is set up so that animals make 1, then 2, then 4, then 8 responses for the first 4 reinforcers. The progressive ratio values continue to increase until animals stop responding. This breakpoint is a measure of motivation, or how hard animals will work for a single presentation of the reinforcer. In contrast to ratio schedules, interval schedules provide reinforcement if a response occurs after a certain period of time. Under fixed interval, FI schedules, the time from the presentation of one reinforcer to the possibility of receiving the next is constant. Under variable interval, VI schedules, responding is reinforced after an average time interval has passed. For example, animals responding under a fixed interval 15 seconds schedule would be reinforced for the first response they make 15 seconds after the last reinforcer. Under the variable interval 15-second schedule, reinforcement would be available on average 15 seconds after the delivery of the last reinforcer. Note that animals must still respond under interval schedules, but they are only reinforced for responses that occur after the interval has elapsed. Situations in which a reinforcer is presented at specific intervals, regardless of the animal's behavior, are called time schedules. Schedules of reinforcement induce different patterns of responding, suggesting that the animal's behavior is determined, at least partially, by its payoffs. For example, acquisition is usually more rapid and declines more quickly under continuous reinforcement than partial reinforcement schedules. This makes sense in that it is easier to form a response-reinforcer association if these always occur together, and the behavior is more likely to cease when the reinforcer is removed if animals were reinforced for every response in the past. Even under partial reinforcement schedules, different patterns of responding emerge. See Figure 4.7 for details of each schedule. Fixed ratio schedules are characterized by rapid responding up to the presentation of the reinforcer. Responding ceases after reinforcement is delivered, and this post-reinforcement pause increases with the size of the ratio. With very high fixed ratio ratios, animals may stop responding altogether, a phenomenon known as ratio strain. Figure 4.7. Cumulative response records under different schedules of reinforcement. The delivery of a reinforcer is marked by a vertical slash. Different schedules of reinforcement elicit different patterns of responding. Fixed interval responding in humans is very accurate if they can use clocks to time the intervals. Bottom line to write a figure. VR equals variable ratio, VI equals variable interval, FR equals fixed ratio, FI equals fixed interval. So on the x-axis is time, and on the y-axis is the total number of responses. So in the shortest period of time, variable ratio gets the highest number of responses. Then after, variable interval gets the second highest number of responses. And these are both linear. Fixed ratio gives you not as many responses over time, and it takes longer. And you can see that it's a stepwise pattern. Okay, and then much later, and you don't get as many responses, you have fixed interval, and it's a stepwise pattern that every time after the reinforcer, there's like a pause, and then at the latest period of time, getting the least number of responses is fixed interval if clocks are used. It's also a stepwise pattern. Fixed interval schedules also induce post-reinforcement pauses that vary directly with the length of the interval, presumably because animals learn that the reinforcer will not be available for a certain period of time. Responding recovers close to the completion of the interval to produce a scallop in responding that peaks when the next reinforcer is obtained. In general, response rates are steady under variable schedules, reflecting the fact that animals do not know when the next reinforcer will be delivered. This pattern can be interrupted by rapid bursts of responding that often occur after animals receive several reinforcers in a row. When the reinforcement payoff is low, i.e. high variable ratio or variable interval schedules, response rates are reduced, responding becomes more sporadic, and pauses occur at irregular intervals. This makes sense in terms of what the animal has learned. Reinforcement is infrequent and unpredictable. In other cases, an animal's behavior appears to be influenced by incorrect assumptions about response reinforcer payoffs. Box 4.2. Box 4.2, Superstitious behavior. The defining feature of operant conditioning is that the presentation of the reinforcer depends on the organism's response. In some cases, however, accidental pairings of a response with a reinforcer will increase the probability that the response will occur in the future, even if there is no causal connection between the two. Skinner, 1948, observed this phenomenon in pigeons by presenting food at brief and regular intervals as animals were moving around a cage. Over trials, the birds developed responses that preceded the food delivery, but these differed across birds. One bird turned in counterclockwise circles, one made pecking motions at the floor, two swung their heads back and forth, one raised its head to one corner of the cage, and another bobbed its head up and down. Skinner surmised that each of these animals had been engaged in a particular behavior, e.g. head bobbing, when the food was delivered, which reinforced the response that preceded it. As trials progressed, the birds were more likely to perform that particular response, which further increased the probability that it preceded the food presentation, and the cycle continued. Skinner described behaviors that were maintained in the absence of a response reinforcer contingency as superstitious. His results were replicated, but then reinterpreted based on a more detailed analysis of pigeon behavior. Specifically, it became apparent that pigeons were able to calculate the time between food presentations, engaging in some extraneous behavior during this interval, and then moving to the food hopper and pecking close to the time of food presentation. Behaviors that occurred during the food interval were often specific to individual birds, which is probably what Skinner was observing. Skinner's findings aside, there is no doubt that many humans exhibit superstitious behaviors, from walking around ladders to searching for four-leaf clovers to wearing a particular piece of clothing at a sports competition. Clearly, not all coincidental occurrences of a response and a reinforcer lead to superstitious behavior, so researchers began to investigate the factors that produce this false belief in humans. These studies concluded that superstitious behaviors are maximized when an infrequently performed response precedes the presentation of a highly valued outcome, and the cost of engaging in the behavior is very low, but the payoff is potentially very high. Even under these conditions, the time and energy devoted to inconsequential responses make superstitious behaviors an evolutionary paradox. One explanation is that causality judgments in humans evolved to take into account incomplete information, and superstitious behaviors are a side effect of this adaptation. Moreover, superstitions are often acquired through social learning, which influences how individuals respond in particular situations. If a young boy never steps on a sidewalk crack and his mother never breaks her back, it helps to confirm his prior belief about this association. Regardless of how they appear, superstitious behaviors in humans are not likely to disappear. After all, why change something that is working? Outside of the laboratory, different reinforcement schedules can be used to shape human behavior. For example, many sales reps receive a bonus for the number of items they sell, e.g. cars or insurance policies, which serves to increase the rate and number of sales. Lottery terminals and casinos are set up on variable schedules to induce high rates of responding with fewer breaks. Gamblers often note that they are reluctant to abandon a terminal after a series of losses because they are convinced that it is just a matter of time until their machine pays off. Unfortunately, sudden high wins help to reinforce this behavior, thereby promoting further gambling. Associative processes as adaptations. Associative processes have been documented in almost every species studied, suggesting that they have been conserved across evolution. Nonetheless, there are many cases in which classical or operant conditioning does not occur, or is not observed according to some predefined measure. Not all of Pavlov's dogs developed conditioned responses, and many researchers are frustrated by the inability to train lab animals in simple operant tasks. In some cases, These discrepancies reflect methodological issues, such as an inability to identify factors in the lab that may influence responding. In many cases, associative learning is slow to develop or difficult to measure because the stimuli and/or responses used in an experiment are not meaningful in terms of the subject's evolutionary history. To appreciate this point, it is important to conceptualize both classical and operant conditioning as adaptive traits, i.e., the outcome of natural selection. Classical conditioning. The adaptive value of classical conditioning relates to the ability to predict significant events. If one stimulus reliably precedes another, organisms can use this information to their advantage. An obvious example is the ability to predict where and when food will be available. This was demonstrated in an experiment with American grasshoppers. Given access to two types of food, one more nutritious than the other. For one group, the more nutritious food was associated with a particular spatial location, taste, and color. The other group had these changed every day. Both groups were able to feed on the two types of food, but insects that experienced the environmental cues paired with the better food group grew more quickly. Thus, the ability to form an association between stimuli and motivationally significant events, i.e., classical conditioning, led to larger, probably healthier animals, which likely provided fitness advantages in the future.